issues seminars. So I was kind of reflecting on this seminar and thinking that, it's just been about 20 years since we started monitoring and studying polyfluorinated and perfluorinated chemicals in our environment. And over the course of that time, it's become painfully clear that these chemicals are quite widespread. I would probably say even ubiquitous in our surface and groundwater. However, the spatial coverage of fish PFAS concentrations remains limited and background conditions remain poorly understood. So today we're going to hear from our three speakers about the work that they are doing to understand the nature and the extent of PFAS contamination in fish across the Lake Superior Basin that is contained within Minnesota, where they have looked at the relationship between PFAS sources and habitat preferences and the level of PFAS burden in fish. Summer Streets has worked as a research scientist at the Minnesota Pollution Control Agency since 2007. She has studied contaminants of emerging concern, primarily PFAS, in the ambient environment for several years. Ryan Lepak is a research limnologist at the US EPA in Duluth, where he focuses on understanding contaminant sources and fate in the environment, especially in the Great Lakes. And recently he's focused on drivers of fish contaminant burdens in the lakes. And last but not least, of course, Michael Mahan is a postdoc research with the US EPA in Duluth. His research is focused on the spatial and temporal patterns of ecological communities in response to environmental change. For example, contaminants, climate change, habitat loss, and his current research is focused on the spatio-temporal patterns of PFAS bioaccumulation in fish in the Great Lakes, which brings us very nicely to the topic of our seminar today. So, you guys take it away. The floor is yours.
Okay, I'm going to start. I'll share the screen, and then partway through, I'm going to turn it over to Mike. And so, Mike, you can just tell me when to flip the channel and all that. But let's see, we're going to talk to you today about a study that was funded by EPA Great Lakes Restoration Initiative. And the purpose of that studies were to go to waters that we had not previously tested, but that had suspected nearby sources. Also to revisit some sites that we had previously tested and we knew had elevated concentrations of PFAS. In addition, we visited waters that are important for harvest, including several tribal waters were monitored for the first time. And the goals of the study were to do general condition monitoring as it relates to PFAS concentrations, initiate source investigations if we found any new sites, and then to update the fish consumption guidance. And one thing I don't have on here that Mike is going to talk about is to use the carbon and nitrogen isotope data to help us understand habitat preference and trophic level bioaccumulation between these different fish. So today we're just going to be looking at the PFAS data and the stable isotope data. The PFAS were analyzed by two different labs, the majority of fish being analyzed by the Wisconsin State Lab of Hygiene, and then a subset of fish, all smelt, collected in 2022 were tested by SGS. We looked at 19 inland lakes, 15 streams plus several sites in the St. Louis River, and then 15 sites within Lake Superior. So far, we've analyzed over 500 fish, and we have another 170 fish that were previously collected in 2021 that are currently being analyzed for PFAS by University of Minnesota in Duluth with EPA in Duluth. Oops, this is not advancing. Why? There we go. Okay, so this is just a list of the different species that we tested, and you can see there's a big range here. Many of them are important game species, but we also have a few non-game species in there, like creek chub, white sucker. So it was a nice range of species that we looked at. Okay, here's a map, dots on the map with the different sites that we sampled. We had several inland sites, particularly around the Duluth area, but also further up the shore, and then multiple sites within Lake Superior. And then if you look at the little red call-out boxes, we've got a couple of different sites highlighted. You can see within those, there are red dots, which means higher concentration of PFAS. And so the smaller box is related to the call-out in the upper left-hand corner of the screen. Those are Fish Lake, Wild Rice Lake that are, excuse me, already on the impaired waters list, and these are impacted by AFFF from the Duluth Air National Guard and the airport up in that region. And then in the other call-out box, we've also got, I think I might have made them backwards. In any case, you'll follow along. We've got Sargent Creek, which was a new site that we found. It is in that upper left box, way down in the corner, there's a black dot. Those were the highest concentrations we saw in any fish, up to over 500 nanograms per gram. And I'll show you those data a little more closely. That's also an AFFF site, but not the Duluth Air National Guard. It's a different AFFF site, and that one is now in our remediation program. So they're working on addressing the issues there. And then you can kind of see throughout the rest of the region, we had very low or relatively low concentrations of PFAS detected and several non-detects as well. So that was good news. Okay, so I'm looking at here basin-wide PFAS by species. These are just the detects, and this is on a log scale so that you can sort of see where the different species shake out. The more elevated species generally came from those impacted sites that I just showed you, with the rest of the species being at that sort of general, it's present, PFAS is present, but at a much lower concentration. Okay, we'll start with Fish Lake flowage. This is a lake we've known about for quite a long time. It's been on the impaired waters list for a while. And just looking at PFOS concentrations in those fish, you can see they're elevated generally above 50 nanograms per gram. There is fish consumption guidance on this water already, along with Wild Rice Reservoir. I don't know if I can move this little box. There we go. Along with Wild Rice Reservoir, which is sort of connected hydraulically to Fish Lake, and it's also impacted by the Air National Guard. And you can see the concentrations there get up to over 200 nanograms per gram. There is advice on that water already as well. And then we also detected PFHXS in the fish, which is another signature of AFFF. Miller Creek, we have two different sites that we tested. These are all brook trout, and quite high concentrations, over 200 nanograms per gram at one of the sites, and even the lower site is relatively elevated. And in those fish, we also found a number of other PFAS contaminants, including some of the longer chain carboxylates. And then finally, Sargent Creek. Let me move this little window here. Maybe I can stick it there. There we go. Sargent Creek, those are all creek chub. We just had 5. We were unable to find other fish in that water body. It is pretty shallow and kind of murky and not really supportive of other fish species. But you can see there the range of concentrations are all very, very high. And so that site was referred to remediation, as I mentioned, and is being addressed currently. We did also in those fish find a PFHXS and PFHPS, which again are good markers for AFFF. Okay, smelt has been of a lot of interest in the last few years, ever since Wisconsin did some testing and found elevated levels in smelt on the south shore of Lake Superior. Mike's going to talk about that quite a lot more. So here I'm just showing you the general sort of concentrations of smelt, which compared to Wisconsin and Michigan are quite low. But it is interesting that we see this pattern of longer chain carboxylates in the smelt. And again, Mike's going to tell you more about that, so I don't want to go into too much detail. Those two little outliers there are the Lester River, which is kind of on the northeast end of Duluth, so in town there, and maybe has more of an urban effect, and that's perhaps why we're seeing those couple of outliers. But in general, the average PFOS concentration in smelt was around 2 nanograms per gram. Okay, I am gonna turn off my camera and my microphone and give this to Mike.
Right.
And let's see if I can do that from here. Sorry, Mike. Well, you just start talking and I'll figure out my view here.
Okay. Yeah, so just to kind of expand upon what Summer was just talking about, this figure here is just another way to look at the map that Summer showed a few slides ago, where each individual bar represents a different sampling location. So water body or sampling location within Lake Superior. In the data that MMPCA collected, and each bar just represents the average of all of the fish for a given PFAS. And so we can see, Summer, if you just want to advance just one, I've got a little box that'll pop up. We can see here that our first 5 sites here are all those kind of known impacted sites that Summer had mentioned, with Sargent Creek being much higher, relative to all of the other sites in the data set. And Summer, if you just want to
why is that doing
advance one more.
that? Is it not? There we go.
And one more too. We'll just do that.
One more.
And so again, we've got here the sites are in the exact same order. So again, they're ordered by the total concentration of PFAS in the system. And our first 5 highly impacted sites are there on the left. And so this is just a way to look at the kind of relative contribution of all of the different PFASs that were found within these fish. And so generally what we see is these first 5 sites tend to have, I mean, all of the sites are really dominated by PFOS, which is really not surprising. But the sites are, we tend to see, especially in the impacted sites, kind of a broader suite of PFAS, and especially those that are associated with AFFF and the longer chain ones that Summer had mentioned. One thing I forgot to mention on the previous slide is just that outside of our kind of 5 impacted sites, the next highest kind of total or average PFAS concentration in these fish is right around 10 nanograms per gram, and every other site is below that. And as you can see here, we got a suite of about 12 sites that all of the PFAS that's sampled were below detection limit, which is really great news. It's, for the most part, outside of these impacted sites, we've got low PFAS concentrations, in the Lake Superior watershed. And that one site that is slightly elevated, outside of the impacted sites is Tisher Creek, which runs right through the heart of Duluth. So again, could be just kind of that urbanization signal that Summer had kind of mentioned. And another way to analyze these data is through the use of an ordination, which Summer, if you just want to advance the slide. An ordination is just a statistical technique that takes multivariate data, so in this case, concentration of multiple PFAS, and compresses that variation just down into 2 dimensions. And so here the units on the X and Y don't really matter. We kind of interpret this kind of like a map. And so points that are closer together are going to be closer together. They're going to be more similar in their PFAS chemical profiles versus sites that are further apart. And the PFAS themselves in red here, those that are closer together are going to be more similar to, or going to be found in kind of the same fish in the same sites than those that are going to be farther apart. And so what we can see here, or if you want to just go ahead, I think once or twice, maybe. Yeah, one more time. What we see here is we kind of get a separation of our known impact sites. And we really do see that AFFF signal here on the right-hand side with those longer chain PFASs. And before I get any further into those, we also see kind of the majority of our sites just coming out right on top of each other, meaning that they've got, no really unique PFAS signature, which is good. There's no, pinpointable kind of PFAS kind of source for those lakes. It's just kind of background level. There is one lake here, Trout Lake, which is at the boundary of the Boundary Waters Canoe Area in the Superior National Forest. And these 3 PFAS chemicals are found in ski wax but they're also found in a lot of different commercial products. And so based off of the kind of geography of where this lake is found and its remoteness, it may be a kind of ski wax signal, but we also don't have kind of the fingerprinting available to say it's for sure ski wax or it's something else. And so, but just something interesting that kind of we weren't expecting. Summer, if you want to advance one more slide. And what we see here is just a zoom up of that ordination to those AFFF and impacted sites. And we can see here, as Summer mentioned, we've got Fish Lake Flowage, Wild Rice Reservoir, and our 2 Miller Creek sites that are all impacted from downstream from the airport and air reserve, and Sargent Creek coming out from the Lake Superior College Emergency Response Training Center. Something interesting is that Miller Creek 2 here, that sampling location, is geographically and hydrologically closer to both the Fish Lake Flowage and Wild Rice Reservoir than this other Miller Creek site. But this other Miller Creek site is showing a kind of a higher relative levels of these other PFASs, potentially suggesting that either these PFAS chemicals are getting kind of reintegrated into the water column and into the fish that are found there, or there may potentially be a kind of secondary or other source of PFAS in between these 2 Miller Creek sites. Summer, go ahead. And so, as Summer mentioned, we also looked at stable isotopes. And unlike other chemical contaminants like mercury and legacy contaminants like PCB and PBDEs, fish characteristics such as age, size, and growth rate may actually not be related to PFAS concentrations within the fish themselves, which can be a little bit of a frustration because you expect these relationships and they just don't hold for the PFAS class of chemicals. And so it can be difficult to actually understand what is driving the PFAS concentrations within these fish. And so factors like habitat and nutrition may be those important drivers then rather than just fish size. And so to get at that, we use stable isotope data. So delta 13C and delta 15N ratios there as a proxy for habitat use and nutritional resources. Now, these isotopes are a kind of integrated measure of these fish. So it's going to be something that accumulates throughout their entire lives. It's not just going to be a pinpointed thing like taking something from the water column, let's say. And so we know that fish move from habitat to habitat within a system, but this kind of provides us a gradient where these fish are spending the majority of their time feeding. And so with this little infographic, we can see that the delta 13C isotope ratio gives us a gradient for the relative amount of terrestrial carbon in a system. So in other words, the distance to shore. So a fish that has lower carbon 13 is going to be feeding offshore versus one that has kind of a higher carbon 13, which would be feeding more coastally. Now, the delta 15N generally also provides a gradient of trophic position, meaning that fish that are top predators are going to have higher nitrogen, delta 15N nitrogen levels. As that kind of food goes up the food chain, it builds on itself. Now, in Lake Superior, this isotope can also provide a benthic to kind of pelagic gradient. So a kind of bottom deep feeding fish versus one that's going to be feeding in the kind of higher areas of Lake Superior, so closer to the open waters in the open areas of Lake Superior. So that's going to come into perspective in the next slide that Summer will move us to now. And so here we use generalized additive models, which basically just says that we allowed the isotopes to not have a direct linear relationship with PFAS concentration. And the gradient here, the coloration, the darker colors to the darker blue to the lighter yellow is just a less PFAS to more PFAS gradient. And in this model, we're also accounting for fish species as well as fish size. That we're taking some of that variation out and this is explaining a lot of the other variation in the model. In fact, it explains 70 to 80% of the variation in the data so far. And so what we can see here, focusing on Lake Superior, on the, inland lakes and rivers and streams here on the left, and in the middle, we have our kind of carbon isotope on the X and our nitrogen isotope on the Y. The X is a pelagic to coastal, once again, kind of gradient. So on the right-hand side are fish that are feeding in or closer to the terrestrial realm. And on the upper part are going to be our higher trophic position fish. So for lakes and streams, we actually see what we generally would expect. We see that we have higher PFAS concentrations along that kind of trophic gradient. So fish that are high, top predators are predicted to have higher PFAS concentrations compared to those that are feeding kind of more basally. Let's see here, make sure my notes. But I want to be clear that this might not necessarily be a biomagnification signal. It could just be that these fish are older, and so it could just be a biomagnification, or sorry, not biomagnification, bioamplification, accumulation, sorry, goodness, bioaccumulation signal, just that these fish have been in the waters for longer compared to the kind of lower trophic fish here. But it's difficult to parse those out, and that is just going to require a little bit more work on our end. The carbon signal is a little bit less clear, but generally we see that fish that are feeding in terrestrial, that have higher terrestrial derived C, have higher PFAS concentrations than those kind of with more kind of aquatic derived carbon. But if you look at Lake Superior on the right-hand side, we actually see the exact opposite pattern. We see that the model is predicting that more PFAS would be found in fish of either lower trophic levels, or remember in Lake Superior, of those that have are kind of feeding more in the open waters, especially those that are feeding we expect those to be feeding closer to shore. These tend to be kind of our rainbow smelt and different types of salmon versus our kind of Cisco at deep feeding lake trout up here. But that relationship is much weaker than our inland systems, and the nitrogen, and as I said there, and so we, as Summer had mentioned, we're adding more Lake Superior fish this winter to gain a better perspective of what is going on kind of in Lake Superior. This set of analysis is very preliminary, and so kind of next steps are to actually scale these isotope data within individual lakes to better account for the lake-to-lake variation, so that way we can look within lake gradients of coastal to pelagic and kind of get a clearer signal there. And so this is a nice transition into what I'm going to be talking about for the majority of the rest of the talk, which is really focusing on Lake Superior. But I'm just going to kind of summarize what we found for the inland systems first. So generally, we find low PFAS levels across the Minnesota side of the Lake Superior Basin outside of those impacted sites. Again, this is really great news. As Summer mentioned, the sampling actually identified an additional impacted site, which is now under remediation. Our PFAS profiles can help to identify impacted sites and potential sources of PFAS contamination, though the fingerprinting still needs to be kind of finalized. And again, across water bodies, PFAS concentrations are greater in fish at higher trophic levels, and those fish that are more proximate to coastal areas. And again, we're still kind of working through those results, so further work will clarify those patterns. So we're going to jump right in now into the cold waters of Lake Superior. We know that Lake Superior is ecologically, economically, and culturally irreplaceable. It represents 10% of the global surface freshwater. There still are about a ton of fish that are harvested annually. And within the total boundary of the watershed, roughly 600,000 people live within Lake Superior. These fish populations are imperiled due to threats from pollution, climate change, and invasive species. So understanding the PFAS impacts on fish will provide clarity to both ecological and kind of the human health side of things as well. Summer, yep, thank you. And as Summer had mentioned, smelt have been of particular interest in Lake Superior for the last few years. with Wisconsin and Michigan finding smelt levels with very, very high, elevated PFOS levels. Now, these are the first PFAS data for smelt in Minnesota. Of 157 smelt, 66 or samples, 66 were individuals, and 91 were composites of multiple fish. For the most part, the PFOS concentrations here, you can see in the orange, tend to be less than 4 nanograms per gram and average about 2. Smelt also found to have measurable concentrations of PFOA, PFNA, and then some longer chain PFASs as well. And generally, it's interesting to see PFOA in the smelt because it's not a PFAS that we find very often in fish in Minnesota, as well as kind of PFNA levels seem to be a little bit elevated. Now, I just wanted to mention that these are whole-bodied fish, and so to compare to headless, gutless fish, the evidence suggests that we can just divide these concentrations by about 2 or 3, so a little bit lower than kind of what is presented here. So Summer, go ahead to the next slide. And so here, similar to the slide I showed a few, or the figure I showed a few slides ago, looking at kind of site gradient of different PFAS concentrations in smelt. Here I've got these ordered by kind of a south to north gradient along the North Shore from Duluth up to Grand Portage. Each bar represents an individual smelt sample, so either a composite or an individual fish. And then the kind of bounding boxes in between each just represent different sampling locations. And so generally we see, and this is all in Lake Superior as well, we see that PFAS concentrations in smelt are relatively low, almost always at or below 10 nanograms per gram. Aside from a single individual collected near the Lester River in Duluth, which is actually just outside my door here at the EPA, had very, very high PFAS, or relatively high PFAS concentrations. And so there may be something to kind of look at with the Lester River in the future. And so despite being located within kind of the same region of Lake Superior as the smelt on the South Shore that had been found to have elevated PFOS concentrations, the smelt on the North Shore of Lake Superior are much, much lower than those that are found in Wisconsin. Next slide, Summer. And so what we can see here, again, looking at the PFAS profile, is that the overall message is just that, PFOS is found pretty much in all of the smelt that in which PFAS was found at high concentration, relative concentration. There's really no site-specific fingerprinting. We can't say that, oh, the Duluth area smelt have a specific PFAS signature. It's just kind of ubiquitous. But again, as I had mentioned, smelt, interestingly, have relatively high concentrations of PFNA in the green, and then 2 longer chain in the yellow and the blue, which is kind of unexpected compared to the other fish that have been collected in Minnesota. And so this suggests that these smelt either have an affinity for the retention of these specific PFAS or are predisposed to these PFAS by habitat preference. Now, PFNA in the green, as I mentioned, is a surfactant. So it could just be that smelt, because they're using that kind of the high portion of the water column, that there may actually just be predisposed to the PFNA, as well as the other PFAS chemicals that are going to be found in that area, because they're surfactants compared to other fish that are going to be feeding lower in the water column. Summer, if you want to go ahead. And so we also wanted to look at, just what might be driving the PFAS smelt concentration. And so again, we have that south to north kind of gradient with an overall superior estimate here. Each individual point just accounts for the length of the smelt sampled. And so essentially just holding all of the smelt to the same size. And what we can see here is generally we see a kind of decreasing PFAS concentration as you get further up the North Shore, so further from Duluth, further from population centers, larger population centers, suggesting that smelt PFAS concentration could be related to the distance from the PFAS source. Now, we also looked at kind of smelt different kind of populations along the North Shore in relation to habitat use. And so what we see, again, on the left-hand side of this figure, we see those are the kind of Duluth smelt versus the Grand Portage smelt. And we see here that the smelt are actually kind of shifting along the North Shore along this kind of 13C gradient from pelagic to coastal. Now, this could mean that there is that these smelt are actually using different habitats within Lake Superior, or there's a carbon-13 kind of gradient along the North Shore of Lake Superior due to changing biogeochemistry along the shore. But this validates that smelt populations are probably different between different sampling locations along Lake Superior and not commingling with each other. And this provides some assurance that smelt are actually consistently low in PFAS along the entirety of the North Shore, and also that those smelt that are found along the South Shore are likely not bringing high PFOS concentrations kind of along to the North Shore of Lake Superior. And so another kind of important game fish in Lake Superior is lake trout. And so lake trout in Lake Superior have two different kind of morphological subtypes. So there's the lean and the Cisco-wet. And the Cisco-wet tend to be found in kind of those are our deep feeding lake trout. But within the samples that contain Cisco-wet morph lake trout up in Grand Portage, there is an order of magnitude difference in the PFOS concentration. So they range from about 2 nanograms per gram all the way up to about just over 20 nanograms per gram. And so we were interested in understanding what might be driving such a broad pattern or difference, broad range in the Cisco-wet found kind of in one site. So if you just want to advance one more slide here. And so we looked at, again, using the isotope data to look at habitat preference. Now, these Cisco-wet all had roughly the same delta N values, and so delta N really wasn't that important here. Instead, what we see is, so if you want to advance one more slide too, kind of a positive relationship here along an increasing PFOS gradient from fish that are feeding more in the pelagic areas versus, so open waters versus those that are found feeding more coastally. So the variability in Cisco-wet PFAS concentrations was not attributable to size differences or trophic levels, as I said. Again, it's really strongly related to habitat use. And this suggests that PFAS may be being exposed, or not, sorry, Cisco-wet may be being exposed to PFOS concentrations or sources kind of in those coastal waters. So something just kind of to think about for future work in Lake Superior. So kind of a segue here into some work that we're doing here at the EPA. And so as kind of has been mentioned and is known about PFAS, is we've really only been monitoring PFAS concentrations for about the last 20 years. And so a limitation of our monitoring efforts is the limited ability to actually establish historic trends and baselines in PFAS concentration going back prior to the year 2000. The Great Lakes Fish Monitoring and Surveillance Program, which was established in the 1970s, establishes 2 sites within each Great Lake where fish are sampled, mostly lake trout, except for 1 location in Lake Erie. Again, the lake trout or the fish are collected, placed into size or age standardized classes, and homogenized. And so, go ahead, Summer, it's fine. And so these samples are retained as kind of freeze-dried samples through time. And so there are about 800 composites representing all 5 Great Lakes. The historic measurements included kind of the best of the time available chemical contaminants. So we're talking PCBs and PBDEs, those legacy contaminants. And because they have been retained, these samples still exist, we can measure kind of the bulk tissue carbon and nitrogen isotopes different amino acid isotopes, as well as going back and measuring PFAS contaminants from these dried samples. And so again, these are dry weights. And so dry weight PFAS concentrations measured on dry weights are usually 4 to 5 times higher than that of wet weights. So Summer, if you want to advance one slide. So some very preliminary data at this point. So we have the Apostle Islands, which represents a shallow site in Lake Superior, versus Quinua Point, which represents a deep site. We have PFAS data going back to the 1980s. And generally what we can see is PFOS in yellow here, kind of showing up right away in the samples and increasing a little bit in concentrations until other PFAS start to kind of show up on the scene, come and go and come and go. And let's see here. Again, what's nice is that we actually don't see any of the other PFAS chemicals appearing in the data set until they're actually put into use and production in the United States. And so again, PFAS concentrations seem to be increasing generally until about the mid to late 2000s. And this is kind of followed by a potential reduction over the last decade of PFAS concentrations in lake trout in Lake Superior. And so these results are promising in that concentrations don't appear to be as high as they have been in the past, but are concerning in that PFOS is still the PFAS chemical with the highest concentrations in fish, despite US production and use being phased out nearly 2 decades ago. One final thing to note is that PFNA, which is also found in high relative concentrations of smelt in Lake Superior from modern day samples, also appears to be in high relative concentrations of lake trout in the last decade or so. And so again, this is really preliminary stuff. We're going to be kind of digging into this data more as it continues to kind of be produced and analyzed, and we'll see some more cool stuff coming out of Lake Superior soon. So Summer, if you want to advance one slide. And so in general, smelt along the north shore of Lake Superior have roughly 4 times less PFOS than smelt collected along the south shore of Lake Superior. Again, this may be due to differences in habitat use and separate populations. The Cisco Wet lake trout PFAS patterns suggest differential exposure histories between those fish that are feeding near and offshore. And the fish archive that the EPA has been collecting and maintains will provide insights into long-term PFAS trends and baselines across all of the Great Lakes. And these the data presented in the previous slide will inform more complicated models, again, establishing trends in chemicals and bioaccumulation and bioamplification, magnification of these chemicals in the fish, with the hope of producing a kind of whole Great Lakes food web model. Summer, if you want to take the last few slides.
Sure. Since I couldn't turn off my camera, here I am still. Okay. So generally, we do see low PFAS concentrations across the Lake Superior Basin in Minnesota, outside of those few impacted areas, which I think in general is good news. Habitat does seem to influence PFAS exposure and resulting fish concentrations. And I mean, obviously, if there's a source that greatly impacts PFAS concentration. It was also good to see in the study that smelt along the north shore of Lake Superior have less PFAS than smelt collected along the south shore of Lake Superior in Wisconsin and Michigan, where they are really seeing quite high concentrations. Perhaps they have a different source or habitat use there, as Mike mentioned. And also some preliminary data suggest that the Lake Superior lake trout PFAS concentrations might be decreasing over the last decade, which I think is another bit of potential good news. I just want to point out that this really has taken a huge number of people to put this study together, and multiple different agencies collaborated on this study. And so thanks to everyone who put in efforts on this. And if you have questions going forward, you could reach out to me via email or phone. And I did just want to provide, before we take questions, one point of clarification on the smelt. The smelt in the study actually are headless, gutless, so don't divide them. There probably are some other whole body ones out there, but for this study, because the original intent was to look at fish consumption advice and look at updating it. We followed protocol that MDH uses and looking at fillets, and with smelt, it's headless and gutless. That is typically what people eat. So just putting that clarification there. So thank you, and we're happy to take your questions. I'm going to stop my slides here.
All right, it looks like there are a couple questions. All right, we have one from Scott. Does Michigan or Wisconsin issue PFAS-related fish consumption advisories in a Great Lake?
Yep, I believe both of those states have consumption guidance, certainly for smelt, possibly for other species, but I don't have the details of that guidance handy. Prevailing wind direction, we do have that. It is right up and down the shore. We have this because we did an air study a couple of years ago, and we had our reference site was in Grand Portage. And so at the time, Dorian Cavalli, who's now at Department of Health, was working with me on that, and I have nice annual average wind rows for the Grand Portage site, and the wind really is up and down the shore.
All right, now we've got questions rolling in.
Okay, so the Siskoet question, and maybe Mike or Ryan can help on this one. Those Siskoet that were collected in this study were collected near shore. And they do seem to look different from other lake trout and Siskoet, even like adjacent, some collected adjacent to that site. So I'm not sure how to respond to that question about living at depths and what do they eat. Perhaps Mike and Ryan have something they want to add to that question.
Yeah, I'm not a fish ecologist, so I can't really clarify. Ryan, do you have any insights as to what those Siskoet might be eating, 250 feet down?
Yeah, so I'm also not a fish ecologist, but I'm an avid fisherman. And Siskoet are cool. So they do a lot of diel migration, and they're kind of wolves. They chase the mysis, but they're gonna anything they can fit in their mouth, they're going to. They love burbot, which are obviously in the benthos, but they also chase these smelt. They chase these mysis plumes, etc. So I think they're kind of getting whatever they get access to. They're not relegated to the benthos.
I've never heard of Siskoet referred to as wolves, and now I have this great visual. Thanks, Ryan. All right, is the lake trout that was tested for PFAS in fish the same lake that is, in trout, is the trout lake that was tested the same one that's in the Sentinel Lakes program? I'm not sure, Sherry. That's a great question. Probably, because I know when we were choosing these lakes some years ago, there was lots of discussion of using Sentinel Lakes and how to choose these lakes. We went through many levels, but I can check on that and get back to you. And Michael Ginsbach put a handy report in the chat. Thank you, Michael. Let's see, how did Tisher Creek concentrations compare to other sites in the study? So Tisher is certainly elevated, not as elevated as like Miller Creek or Sargent Creek or Fish Lake or Wild Rice Lake, but the fish are elevated. And I want to say the concentrations are around like the mid-20s nanogram per gram. That's kind of what comes to mind. We don't know what the source is there. Jesse Martis and I have been talking about that pretty extensively, about trying to get additional funding to try and do some reconnaissance type sampling and see if we can pinpoint a source. But we're not sure, but they definitely are elevated above sort of the other inland lakes or the Lake Superior fish. Okay, were PFAS concentrations sampled in the water? No, I'll just say that right away. And could that be one explanation? I guess I'm asking if the water concentration PFAS could be well, certainly it could be, right? But typically you need something beyond like an atmospheric source to get those really elevated levels. So when we see like Fish Lake, Wild Rice Lake, Miller Creek, Sargent Creek, those sites also have elevated water concentrations. And if you track back to the source, those water concentrations go up and up and up and up. Same with the sediment. If we were to take water from one of the lakes where we don't have any detections of PFOS, for example, I would bet dollars to donuts that you're going to get non-detect for PFAS. You don't need very much PFOS in the water for there to be a detectable amount in the fish. But you're not, that said, you're not going to get these super elevated fish concentrations without some kind of ongoing source that is direct. We just don't see very high levels all over the state. I mean, I've been doing this fish work for a long time, and you just do not see the very, very elevated concentrations without some kind of current ongoing source. Any thoughts on PFAS affinity for microplastics? Or maybe Mike or Ryan could reply to this as well, but I think in general, what's been seen is that microplastics have an affinity for all sorts of hydrophobic organic contaminants, and PFAS can also be incorporated into plastics, so I would not be surprised if there's some type of connection there, but do I have any better, nothing more to say on it than that, I guess. I don't know if Mike or Ryan have any thoughts.
I agree.
All right, well, I don't see any other questions in the chat.
I have one that I would like to follow up on, Summer. This is Mark again. Just to follow up on the question about the precipitation source. One of the things that I'm suspecting more and more is that other emerging chemicals of concern, as we call them, one of the things I'm starting to suspect is that their presence at remote locations can really be due to atmospheric fallout, whether it's precipitation or particles. And while you were saying that the concentrations you were seeing in fish can't be attributed to that alone, I am really curious if you guys have any insight to what, I don't know, for lack of a better term, that background introduction of PFAS from precipitation and particulates might account for some of the results you're seeing. I mean, I'm kind of curious about that.
Well, I'm certain that there is an atmospheric impact, right, to some extent. And I can say that with certainty because we have a whole year's worth of atmospheric deposition and air data from Grand Portage. We have a whole entire year's worth of PFAS data for that. And what was interesting and surprising about that was we selected Grand Portage as a reference site because it's relatively remote. There's really not a lot of industry right there. And it turned out that those air concentrations for PFAS were second only to the St. Louis Park concentrations, where that air sampler was located right next door to a chrome plater that we know emits PFAS in air. So it was surprising. It was very unexpected to see that. So and also just to add on to that, Wisconsin has been doing a lot of work on PFAS in precipitation and dry deposition. And certainly it's there, but concentrations are pretty low. They're in like the single digit nanogram per liter in precipitation. So certainly there's a contribution. And you can maybe see that in waters where there isn't a direct source and you'll have a little bit of PFAS in the fish, but it's not ever, in my experience, going to amount to the 500 nanogram per gram type concentrations that you see in like Sargent Creek, for example, right? So there'll be that kind of low level, couple of nanograms, maybe, but not super high. Does that help?
Yeah, I think that's great insight, actually, and very interesting data from the Grand Portage site.
Yes, perplexing. And I've had a lot of conversations about that site, and who knows what's happening there. And it's interesting too, because then you have those Siskoet that were caught in Grand Portage that look different from, the fish in the rest of the lake that we collected anyway. And so it does beg the question of, is there some kind of source there that we're just not able to pinpoint at this time? How do you respond to questions about people concerned about swimming and wading? Luckily, I don't generally have to respond to those questions. We usually direct them to the Department of Health, but in general, particularly in these waters where you have, no PFAS detectable in fish or very low concentrations detectable in fish, I would absolutely tell people they're safe to swim in without any hesitation at all. So in general, I think most of our waters are probably safe for swimming. As far as some of the higher concentration sites, even like the Fish Lake and Wild Rice Lake, those water concentrations are not going to be very high, like higher than the background, but they're still going to be in like the tens of nanograms per liter, which is probably not any kind of issue for swimming. So generally, I think swimming is all right. We usually tell people to avoid foam. I think that's been the line primarily from Department of Health and NPC as well. Like if you see foam, just leave it alone because that could be elevated in PFAS. But otherwise, swimming in most areas is probably safe.
You can also feel free to unmute if you would like to be brave, ask a question, or put it in the chat, either way. Otherwise, if nobody has anything else, we're at 5 to 10, so we can certainly wrap up if there are no other questions. All right, well, thank you all for your wonderful talk. It was extremely interesting. One of the highest attendances I think we've seen in a while for water issues. So yeah, thanks so much for presenting for us today. We really appreciate it.
Well, thanks for having us. It's my pleasure.
Yeah, it's been just a really great talk. Thanks for the great seminar.